Today we will learn about kingdom fungi. The kingdom fungi is also called as multicellular decomposers and their study is mycology. The earliest approach was done by scientist Clausius and this name fungi was given by Gaspard and Bauhin. Then afterwards the father of mycology, which is a study of fungi as well as plant pathology, was Anton D. Berry. When we talk about the thallus organization or body design, see that the plant body is for true fungi, that is Eumycota, is thallus, and it contains mycelia, which is in another term called as flagella. Now these mycelial forms, the body also contains thread-like forms which are called as hyphae. So here these mycelia form the hyphae. When we talk about some of the uses of fungi, so this yeast is used to make bread and beer. Similarly, Puccinia causes the rust disease in many plants and penicillin is used to make antibiotics. These three are fungi. Now when we talk what is the network of hyphae called, the network is called as mycelium. If the mycelium cells have no separation, that is they aseptate, then they are called as cynocytic. That means that there is no separation between the cells of mycelia, which are actually the flagella or the network type of hyphae. Then if they have cytoplasm fused, because if they have no cell boundary, their cytoplasm might be fused and that condition is called as cynocytic. If they are septate, this is another case. In some fungi, both stages are also present, where at one stage they are septate and another stage they are aseptate. This is called as dimorphic. The cell wall is made of chitin and polysaccharides. Here the fungi cell wall is not made of cellulose or pellicle. Here most fungi are heterotrophic in nature. That means they are not autotrophs, they depend on other organisms. Of the fungi also live in the symbiotic association like lichens and mycorrhiza. If the fungi associates with algae, it is called as lichens, and if the fungi associates with the root of higher plants is called as mycorrhiza. Here the reproduction in fungi is carried out by three major processes: vegetative, asexual and sexual. The vegetative propagation involves fragmentation, fission, budding. Asexual reproduction involves spore formation, which are called conidia, sporangiosperms or zoospora, and the sexual reproduction, which include oospores, ascospores and basidiospores. The spores produce distinct structures which are called as fruiting bodies, and the steps which are involved in the sexual reproduction. First one is the fusion of protoplasm, that is the nucleus and its contents between the motile or non-motile gametes, which is called as plasmogamy. The next mechanism process is fusion of the two nuclei, that is karyogamy, and afterwards the meiosis in the zygote forming the haploid spores. The mechanism of sexual reproduction we will be discussing now. As you can see that when the fungus reproduces sexually, the two hyphae which are haploid, if they are of compatible mating types, then they come together and then they fuse, which results into the formation of the diploid cell, that is 2N. In some cases we see that only like in Ascomycetes and Basidiomycetes there is an intervening dikaryotic stage where the formation of zygote does not take place and the cell is having two nuclei, that is N plus N composition, and this is called as dikaryon, and this phase is called as dikaryon phase of the fungus. Afterwards the parental nuclei fuse and the cells become afterwards diploid, and then afterwards reduction division occurs leading to the formation of haploid spores. The morphology of mycelium, mode of spore formation, fruiting bodies form the basis for the division of kingdom into various classes.